For the past 5 years, I've studied about breast cancer, specifically the triple negative breast cancer or the TNBC subtype. The initial reason why I started my research was because right before I entered middle school, my mom sadly got a breast cancer diagnosis. And at the young age, I wanted to make sure she 100% fully recovered as she was my only parent, and I thought the best way to do this was to study about breast cancer. Although I first started my Cure breast cancer projects in order to help my mom and make her well, as the years slowly progressed, I started to sympathize for not just my mom, but the entirety of breast cancer patients. During the past several years, while completing these Cure breast cancer projects, I pondered on what the word cure could mean. At first, I believed that a fast diagnosis and inhibiting metastasis was the way for the cure of breast cancer to happen. However, after I found out that there was no cure for metastasized breast cancer patients, I later came to an understanding that to give the present of a cure to breast cancer patients, I needed to find a way to completely treat or inhibit metastasis or relapse. Many papers state that targeted therapies significantly increase distant metastasis-free and relapse-free survivals. Therefore, my focus shifted to treating aggressive breast cancers without a targeted therapy. For this year's project, my main inspiration came from the past 5 years. Breast cancer can be characterized into four main subtypes, as you can see here, dependent on their expressions in the estrogen and or progesterone levels, or the amplification of the HER2 gene. Among the different subtypes, the hormonal positive breast cancer boasts the highest survival rate, while TNBC is the most aggressive and has the worst prognosis among all of them. While checking the gene expressions in different breast cancer subtypes last year, I found that genes highly expressed in this most aggressive TNBC subtype were also highly expressed in the ER-negative PR-positive hormonal positive breast cancer. This was very intriguing to me because hormonal positive breast cancer, as I mentioned before, is known to have the best survival among the different subtypes, and yet one of its subcategories had highly expressed genes of the most aggressive breast cancer subtype. So, searching for papers based on this interesting result, I found that tamoxifen, a targeted therapy that remarkably raised hormonal positive breast cancer patient survival rates, was not at all effective in treating the ER-negative PR-positive breast cancer. And this was because tamoxifen mimics the role of the estrogens and binds to the estrogen receptors. However, the ER-negative PR-positive hormonal breast cancer has no estrogen receptor expressions. And furthermore, continuing my research, I was able to find that there were currently no targeted therapies for the ER-negative PR-positive breast cancer patients. And furthermore, the 10-year survival rate of the ER-negative PR-positive patients were lower than that of triple-negative breast cancer patients by 14%, according to the March 2010 Journal of Breast Cancer paper. This led me to develop my two main hypotheses. First, the ER-negative PR-positive breast cancer is characteristically more similar to triple-negative breast cancer. And two, the inflammation inhibitor, which I newly found as a TNBC target therapy last year, might be effective as the first targeted therapy for the ER-negative PR-positive breast cancer patients. To check my hypotheses, I separated my experiments into three main categories. First, the gene and clinical database analysis, second, the in vitro work, and third, the 3D model. The analyzing of 53,805 genes based on breast cancer subtypes and gene expression analysis in each breast cancer subtype using 4,319 breast cancer patient samples have been continuously done for the past 5 years. And through the analysis of 1,268 genes highly expressed in only HR positive or hormonal breast cancer and 552 genes highly expressed in only triple negative breast cancer this year, I found that the gene expressions in the ER-negative PR-positive patients are mostly similar with triple-negative breast cancer patients than that of hormonal-positive patients. In the first year, I was able to find that CDC20 was highly expressed in TNBC. The second year, I found that included among the main pathways of triple-negative breast cancer was inflammation-related pathways. Last year, I found that pyroptosis-related inflammation genes were highly expressed in triple-negative breast cancer. And at the time, I found that triple-negative breast cancer cell growth and metastasis was regulated by the USP1, CDC20, and caspase-1 axes. And using these past 4 years of my TNBC research as a base, I found that The genes I found last year, or CDC20, USP1, and caspase-1, were also highly expressed in the ER-negative, PR-positive, hormonal-positive breast cancer subtype. Furthermore, when completing the knockdown of each specific gene or adding an inflammation inhibitor or the ACY-BAT-CHO, I found that the ER-negative, PR-positive hormonal breast cancer cell growth and metastasis were inhibited through the completion of colony-forming, wound healing, and Boyden-Chamber assays. And all experiments were conducted at the Mayo Clinic late at nights or during the evenings following the COVID restrictions placed in my town. Through my research, I was able to find for the first time the characterization of ER-negative, PR-positive hormonal breast cancer to be more similar to triple-negative breast cancer than it is to the HR-positive breast cancer, and the first novel target therapy for the ER-negative, PR-positive hormonal breast cancer. I believe that my research will be beneficial to the breast cancer patients who doesn't have a targeted therapy available to them until now. Additionally, my experiment was completed using the 3D culture system, which shows the closest tissue-like structure to human tissues and a high clinical relevance compared to other study methods such as the 2D cell culture and animal experiments. Furthermore, using the 3D culture system, they form spheroids, which is very similar to what an actual cancer looks like in the human body. Therefore, my findings have the great potential to become an actual targeted therapy for breast cancer patients. Furthermore, I would like to explain the potential of my project in the field of cancer research, not only in breast cancer. I was able to find for the first time that CDC20, commonly known as a mitotic regulator, was a key regulator of pyroptosis. Now, pyroptosis is a newly found programmed cell death such as apoptosis, necroptosis, or autophagy. And many papers state that pyroptosis is an inflammatory cell death. Until now, many studies have focused on pyroptosis and its tumor suppressor functions. However, recently, several papers suggest the role of pyroptosis in oncogenic functions. And they further suggest that depending on which genes are highly expressed, this can completely change whether pyroptosis has oncogenic or tumor suppressor functions. I am suggesting through my research that CDC20 might be that modulator for pyroptosis. Pyroptosis-induced oncogenic activities show a very close correlation to aggressive cancer progression as shown in a 2019 Cell Death and Disease paper. Therefore, my research can not only be applied to breast cancer, but it could be the solution to inhibiting aggressive cancer progression, including cancer metastasis and invasion. And additionally, since the candidates that I found through my research are not or lowly expressed in normal cells, I expect that there will be less side effects. And among the side effects, one of the biggest problems currently are liver injuries, and these liver injuries are caused by inflammation. Therefore, the inhibitor in my study will be hitting two birds with one stone, anti-cancer effects and minimizing the liver injuries through anti-inflammation effects. In my study, there were several people that helped throughout my entire project. I would like to thank my main mentor, Dr. Lee. And next, I would like to thank Dr. Lowe for supporting the lab and equipment, mentioning that I could use them whenever and wherever I needed them. And finally, I would like to thank Dr. Ruddy and Leon Ferre for helping me with the clinical aspects of my cure of breast cancer research. They helped me within a day after I sent them an email, and I was very thankful. And finally, the last person I have to thank is Zach, who is my advisor from the Jack and Cook Foundation, for consistently giving me emotional support and cheering me up when I felt down about my projects. I submitted my project that I've been doing from 2016 to a journal or an SCI journal called Breast Cancer, with its impact factor of 2.695. Thankfully, I was able to receive a revision for this manuscript, and recently I was able to finish the revision, and I'm currently waiting for their response. And I'm also preparing for submitting another paper in the summer. And this topic was thankfully and graciously accepted for the poster presentation at the American Association of Cancer Research, which is the biggest and worldwide cancer conference. Finally, my projects have been chosen as the ISA finalists for the 2 years in 2020 and the upcoming 2021. This year, my project was advanced also to the state level for the JSHS competition. Thank you, Davidson, for reading through my application and watching my video, but more importantly, for giving students a hope and a dream. I hope that I can get a good result through this competition so that this research can be shared with many others. Thank you again.